Welcome to my gut microbiome lab. A microbiome refers to the types and the proportions of microorganisms that are present in a given location. So therefore, the gut microbiome would be those proportions and types found in one's gut. There have been numerous correlative studies that have shown the effects that microbiomes have on obesity and one's gut health, how it affects the development of IBS. So our question that we were exploring for this lab was whether or not age and health will correlate when examining the gut microbiomes of an animal. And our hypothesis was that we predicted that the younger animals' over health will be better than the older animals. So for week 1, we used the Keigan kit to purify the DNA. The kit was used to isolate DNA from the sterilized fecal samples, and we also used the Zymo kit, and we followed the instructions exactly as they were given to us for both, but we were using the Zymo kit to concentrate the DNA from our samples after purification. Our original samples that we were working with didn't end up yielding enough data, so all of the results you'll see later are from samples from previous labs that worked, and those were of a 6-month-old dog and a 4.5-year-old dog. On week 2, we were doing DNA quantification. That was done to quantify the isolated DNA and try to get an estimate of how pure it was. And to do that, we used the nanodrop spectrophotometer. We also did our amplicon PCR reactions, and the purpose of that was to amplify the 16S ribosomal RNA gene, specifically focusing on the V3 and V4 regions. So here's our results from that. First, the DNA was set to a temperature of 95 degrees Celsius, and that was to allow it to denature. After that, it was set at 72 degrees to extend the DNA, and then 4 degrees to store the products once it was done, once there was enough cycles that produced enough copies of the DNA. So for week 3, we used the gel electrophoresis. We added the DNA into the agarose gel, and the smaller DNA traveled further down the gradient. That was set at 250 volts. So the purpose of this was to check the success of the amplicon PCR reactions. Next, we did PCR cleanup, and for that we used the Agincourt Ampure XP, and we followed those instructions exactly as they were given to us from the kit. The purpose of using this Agincourt Ampure XP kit was to clean off all the impurities, primers especially, while keeping the DNA sample intact. If they weren't removed, they would interfere with the next steps in our lab. So after that, we did index PCR reactions, and the purpose of that was to add a set of short sequence tags to the ends of the amplicons from each sample, and that just helps us identify which sequence came from which samples. They're easily identifiable by the unique tags that attach to them. Week 4, we did PCR cleanup, and we used the Agincourt Ampure. We followed those instructions exactly as they were given, and the purpose was to clean the impurities off, as stated before. We also did quantification of the DNA indexing PCR products, and for that we used the fluorometer rather than the spectrophotometer because it's far more accurate and it requires a much smaller sample to yield results. We also did DNA sequencing, and we used the Illumina MySec Next Generation DNA analyzer to perform that. Here are results from the spectrophotoscopy. Here you can see how far our samples traveled in the agarose gel. This section right here was the only result that we yielded. That was from our sample 639. I believe that was a chicken. We also yielded results from our positive control, and our negative control also worked, but didn't show up, of course. Here are results from the fluorometry. As you can see, our positive and negative yield results, as well as only sample 639. So these were the new samples that we were using from previous labs that we chose to compare. Sample 307 is the younger retriever, and 308 is the older retriever. And as you can see, we got far more reads for the older animal. So this table shows the reads that we've got for each level of classification right here. So for kingdom, we saw that the older dog had far much more bacteria found in it than the younger dog, as well as the older dog had a virus present. So that just shows that the younger dog's health was a little greater than the older dog's, simply because the younger dog had no viruses found. Here is a table showing order. Here is the table for genus. Unclassified simply refers to the microbiomes that do not belong to any class. So that's what that column up there would mean. Oh, and then other refers to the sum of all classifications less than 3.5% abundance. So for our discussion, we were only able to extract DNA from one of our samples, 639. The rest of our samples, either there was no DNA present or there just simply wasn't enough to get any reads for it. There may have also been an error with the UV spectrophotometry. The concentration we extracted from the sample was 51.2 nanograms per microliter. The ratio of absorbance was 260 nanometers over 280. It was 1.59, which is an acceptable range because the ratio should range from 1.5 to 2.2, and since it was 1.5, it meant there was very little contamination. So therefore the DNA sample would be pure enough. The sample had high enough concentration that the UV spectrophotometer was able to determine concentration of the DNA sample from the scat. A reason for the lack of obtained DNA could have been that the quality of the samples we had chosen. For amplicon, the PCR results, the negative control had an absence of PCR product at about 550 base pairs. This is because the negative control had no DNA added. Therefore, it just confirmed that there was no cross-contamination in our samples. This was expected because we did replace the DNA with water. For the positive control, we used the culture E. coli, which is everyone's very familiar. So we did yield results from that, and that was used as a comparison between the samples and our known sample of DNA. Based off the positive control, it can be concluded that the reaction did work because there were bands from a sample at about 550 base pairs. For our sample 639, the concentration of DNA product was 51.2 nanograms per microliter, and the positive control had about 47.9 nanograms per microliter. Even though the minimum concentration needed was 4 nanometers, the concentrations were too low to be sufficient. Only one of our samples were truly sufficient. For the negative control, the value showed it was too low to read, which makes sense because there wasn't supposed to be any DNA in the negative control in the first place. So, as I stated before, that just supports that there was no cross-contamination in our samples. So the number of total reads for the positive control was around 15,000. In addition, the percent reads passing quality filtering was about 100%. This meant that it worked because we received 100% of the reads. The type of organism identified at the kingdom level were bacteria, unclassified, and viruses, and the virus, as I said before, was present only in the older dog. The species tested levels contained unclassified, bacteroids, Clostridium. The unclassified present is a species of bacteria in the same genus as E. coli, and because of this, it can be concluded that there was little cross-contamination at the kingdom in our positive control because we were 99.9%, almost 100% bacteria. At the species level, there may have been contamination due to other forms of bacteria present. Our sample 307 had a total of 2,687 reads. Everything was 100% successfully read. Sample 308 had 8,028 total reads, and everything was 100% successfully read. Our numbers were adequate to base our analysis on this because they were all in the upper thousands. Dog 307's kingdom is 75.2% bacteria and 24.7% other. Dog 308's kingdom is 99% bacteria and 0.98 other. The dogs aren't similar at the kingdom level, and they differ in the other category as well. This difference is most likely due to the genes knocked out, though there is no way to determine which gene is responsible. Dog 307's order consists of bacteroidales, unclassified, clostridials, fusobacterials, and other, and Dog 308's order consists of bacteroidals, unclassified, clostridials, fusobacterials, and other. The dogs also differ greatly at the order level. Noteworthy differences include that Dog 307 had far more bacteroidals and has flavobacterials than Dog 308. Dog 307 and 308 have many classifications in genus, and because of this, only 7 will be analyzed. Dog 307 has unclassified, bacteroids, Clostridium, and other, and Dog 308 has bacteroids, unclassified, fusobacterium, and other. Dog 307 had Clostridium, and Dog 308 did not. Dog 308 has the same major bacteria. The difference in genus classification would be most likely due to the gene modification in the dogs, though we're unable to say which gene. So back to our question, whether or not age and health will correlate when examining our gut microbiomes. We hypothesized that our younger animal's overall health would be better than the older animal, and our hypothesis was supported by our results because comparing Dog 307 and 308, several classifications of genus were recorded. However, there is no way to determine if gene affects this, which means more research is needed. Also, as I stated before, just the fact that the older dog, 308, had viruses present just shows simply that the younger dog is healthier.